Human Genome Project. It is an international scientific research project with the goal of determining the base pairs that make up human. Goals of SGP include: first point, identify all the approximately 20,000 to 25,000 genes in human DNA. Second one, determine the sequences of the 3 billion chemical base pairs that make up human DNA. Third point is store this information in databases. Fourth point, improve tools for data analysis. Fifth one is transfer related technologies to other sectors such as industries. Sixth point, address the ethical, legal and social issues called LC that may arise from this project. So, Human Genome Project is a mega project. Now let us look the salient features of human genome. First one is the human genome contains 3,164.7 million nucleotide bases. Second one, the average gene consists of 3,000 bases with the largest known human gene being dystrophin at 2.4 million bases. Third one is less than 2% of the genome codes for proteins. So note all these points and try to learn well. So this is the double helical structure of DNA. And the fourth point is the functions are unknown for over 50% of the discovered genes. Fifth point is chromosome 1 has most genes, that is 2,968, and chromosome Y has the fewest, that is only 231. Sixth point, repeated sequences make up a very large portion of the human genome. Hope you understood all the 6 points well. Fifth point, once again note it, it is very important. So this is the emerging field of research also. Now the scientists have identified about 1.4 million locations where single base differences occur in humans. Methodologies of SGP involves 2 major approaches. First one is focused on identifying all the genes that are expressed as RNA, referred to as expressed sequence tags. And second one is sequence annotation. This blind approach involved sequencing the whole genome, both coding and non-coding, and later assigning functions to the different regions. For sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively smaller sizes and cloned in suitable host using specialized vectors. So the cloning resulted into amplification of each piece of DNA fragment so that it subsequently could be sequenced with ease. The commonly used hosts were bacteria and yeast, and the vectors were called the BAC, bacterial artificial chromosome, and YAC, yeast artificial chromosome.